Video is on.
So just give me a minute, sir. Checking it out. No, sir, your face is not yet visible. You can turn off your camera and you can turn it on back, sir.
Done 3 times, same thing. Can I sign out and sign in again?
Yeah, you can do that. You can do that, sir. Okay. 1 minute to go, we can have.
I think it will come automatically after some time.
Okay, sir. So with all your due permission, sir, can we begin? It's 4 o'clock already.
Yes.
All right. So thank you so much. Good afternoon, everyone. This is Badal from IGCP. And I would like to welcome all the viewers who have joined our platform for the better exchange of knowledge that is going to be shared by our keynote speaker doctor for today's session. He is none other than a very renowned HIV specialist, Dr. Ketan Ranparia, sir. Sir is HIV/AIDS consultant at HIV/AIDS Clinic, Surat, Gujarat. With the help of sir, we'll be taking a close look on today's topic, which is how close or far from HIV vaccine. On this occasion of World AIDS Vaccine Day, we'll be discussing more about the topic in details. Before moving on to today's topic, let us have a brief look on what the topic says. For decades, scientists and researchers have been working tirelessly to develop a vaccine for HIV, the virus that causes AIDS. Despite significant progress in HIV treatment and prevention, there is still no effective vaccine available to prevent HIV infection. However, recent advancements in vaccine research have raised hope that an HIV vaccine may be within reach. Now, on this occasion of World AIDS Vaccine Day, we'll be learning more about the topic with Sir's wonderful input. So now, without any further delay, I would like to welcome Sir at our forum and would like to hand over the session to him for his talk to begin. So over to you.
Thank you. Today's topic is how close or far from HIV vaccines. HIV Vaccines Day, it's also known as a HIV Awareness Day. Today is the 18th May and we are today celebrating observing the World's HIV Vaccine Day. It started since 1998. Why this day we are observing? We are observing this day for acknowledge and thanks the volunteers, community members, health professionals, supporters, and scientists who are working together to find HIV vaccines and also create awareness among the community how HIV vaccine is important to prevent this HIV. Why is there no licensed HIV vaccine till date? Especially there are 4 reasons. First is a lack of natural immunity to HIV. Like measles, in the case of the measles, once persons got the measles infections, the body developed immunity, and in most of the cases, that person will not develop measles in their lifetime because our body develops natural immunity to measles. But the same things not happening with HIV. So there is a lack of natural immunity to HIV. Other reason is variability of HIV type. As such, it is a one virus, HIV virus, mainly two type, HIV-1 and HIV-2. There is subtypes N, M, O, P, Q, many. But if we see the total number of the virus as per the structure of different virus, then there is a more than 5 crore different kind of the structural different viruses there. So there is a huge variability one virus for one virus to other virus. Third, lack of correlate of the protective immunity. We don't know, even if we develop the immunity for virus, that immunity can helpful for us in the future for how many months or years. And fourth and last, there is a lack of animal models that can predict the vaccine efficacy in the human being. In 1981, CDC, that means Center for Disease Control, reported first HIV case in this world. In 1987, first time vaccine trial in chimpanzee and 81 MSM. That means men who have sex with men. In 1990, first time vaccine research reached till phase 3 trials. I would like to mention that there are 3 phases for developing any vaccine. And before that clinical phase, there is a preclinical stage that in the laboratory. And after that, phase 1, in that less than 100 persons participate. In phase 2, 100 to 1000. And phase 3, more than thousands human beings participate for checking the effectiveness of HIV vaccine or any other vaccine. In 1994, first time the broadly neutralizing antibody developed. What that does mean? Previously, there was a strain-specific antibody was developing, but problem with that strain-specific antibody was that antibody can act against only particular kind of the HIV virus, but the same antibody cannot affect or control the other kind of the HIV virus. But after developing the broadly neutralizing antibody, there is a hope that one kind of the antibody can control or act against many different kind of the HIV virus. In the 1997 May 18 speech, US President Bill Clinton announced the national goal to develop an AIDS vaccine within a decade. And the day is then known as a World AIDS Vaccine Day. 1998, phase 3 efficacy trial enrolling the 5,400 volunteers. First time the large number of the volunteers was participating in the clinical trial, and mostly was MSM, men who have sex with men, and especially in US, Canada, Netherlands, and the Puerto Rico. In the same years, another phase 3 trials was started. That was in the Thailand, and approximately 2,500 IDU, that means injectable drug users, was participating. In the 1999, first times the African countries, Uganda host the first HIV vaccine trial. In the 2000, surprising results from the study RV 144 trials, and the first time to show any evidence that broadly neutralizing antibody vaccine can protect against HIV. Again, I am explain what is the difference between the strain-specific antibody and the broadly neutralizing antibody. Before, scientists was trying to develop the antibody that was helpful to against only particular kind of or particular strain-specific antibody. But later, when we fail in the previous attempt, scientists developed a broadly neutralizing antibody. That means the one kind of the antibody can act against many kind of the HIV virus. In 2003, RV144 prime boost trial begins. That was in the Thailand, and 16,000 person was participating in the trial, and mostly was a low risk volunteers. In 2009, RV144 results show that vaccine candidate reduced the risk of HIV infections by about 31%, and providing the first evidence of vaccine efficacy in human being. 2021, Moderna HIV vaccine trial started. And 2023, this year in January, Johnson & Johnson's Mosaic vaccines failed, and that was in the only vaccine in the phase 3 currently. But that vaccine also stopped due to the ineffectiveness of vaccine. Currently ongoing, only 4 preventive vaccines trial is there. 2 from the Moderna, 1 from the Vir Biotechnology, and the 4th one from the Worcester's HIV vaccine. So why we need a vaccine? Because currently whatever preventive measures are there that are not effective to control the spread of HIV virus. Currently we are using PEP, post-exposure prophylaxis, condom, PrEP, that means pre-exposure prophylaxis, circumcision, and TAP, that means treatment as preventions. Even after combining all these measures, we are not able to control the HIV spread. And currently, 38 million people are living with HIV. Every year, around 8 lakhs people are died due to AIDS. And every year, 1.7 million people are newly infected. Until date, no infectious viral disease ever been eliminated without use of vaccines. That's why we also need vaccine for HIV. These are the phases for developing any HIV, any vaccines. First, have to develop the molecules or the formula in the laboratory. That same has to test in the laboratory animal. After that, phase 1 that lasts for 1 year. After that, phase 2 that lasts for 2 years. And after that, phase 3 that lasts for 5 years. But all the times, phase 1, phase 2, and phase 3, all the clinical trials must be approved by authority. Then and then only we can move from the one phase to another phase. What we have achieved for other infectious vaccines? We took 33 years for develop the HPV vaccines. We took 35 years for yellow fever vaccine. We took 46 years for measles vaccines. And we took 47 years for polio vaccines. Till date, in more than the 20 years or 30 years trials, only 6 vaccines can reach the third trials phase 3. Mainly 2 kinds of the HIV vaccines we are trying to develop. First is the preventing, that means to control the and preventing getting the HIV virus into the HIV negative person. And the second type is the therapeutic vaccines. That's for those who are already infected with HIV virus. To control the infections and to control the problems using that vaccines, and that known as a therapeutic vaccine. In today's presentations, we will discuss in details about the Moderna HIV vaccine trial that is going to use for the prevention, that means for HIV negative person. That is mRNA based vaccines. In these vaccines, part of the DNA or the RNA will be used. There will be no use of any live virus. So there will be no risk of getting HIV from the vaccinations. And this technology is easy compared to the other kind of the technology. And by this technology, we can produce very large number of the vaccine doses in the very short periods, and the same technology used for the COVID vaccines. First trial, phase 1, was started in the America. 56 healthy individuals, 18 to 56 years of age, those who are free from HIV and has a low risk behavior, and 1 to 2 vaccines dose will be given. Clinical trials started in the September 2021, and that will, how that vaccine will help. That vaccine will be developed by using the beta cell that will produce broadly neutralizing antibody, and that attack and invade outside of the virus. I have already discussed what is the difference between the strain-specific antibody and the broadly neutralizing antibody. And first part, the 2021, that study known as a IAVI-G001, that was a HIV-specific antigens and target the beta cell immune response, and that was developed. 97% of the vaccine recipients developed the immune response. In G002, it was in the 2022, same, it was in the US, and it was a second phase of the trial, second part of the phase 1. And now, international AIDS vaccine initiatives associated with this Moderna vaccines trial. In G003 study, now it's in the Africa, and within 1 or 2 months, we can expect the outcome of that study. Previously, only one kind of the antigen was used, but later, another kind of the antigen added in the vaccination. And this African trial, 18 healthy HIV-free adult volunteers was involved. 2 doses was given, and that during this vaccination trials, the portions of the viral sequence used not live virus. For that, there is no risk of getting the HIV due to the vaccination. It will take 6 months, and what that will study the effectiveness of the vaccines and the how safe that vaccine is. Result will arrive in this 1 or 2 months. We can expect that these vaccines will be safe, effective, affordable, and the immunity can protect us from the HIV for many years. So when will vaccine arrive? But still, it will not come before the 2030. We can accept that if everything goes as per the scientist plan, and we will not get any failure in between. Even then, we require at least 2030 times to reach and get the HIV vaccines in this one. Any questions? If you have any questions, you can type or the mentions in the comment sections or the
That's all from my sides for today's lecture.
So the question is in the chat box.
Okay. What are the challenges in the development of HIV-1 vaccines? Okay, I have mentioned 4 things. First challenge is that HIV virus is not a stable virus. Coronavirus was a stable virus, so we could get the vaccines for the corona. But the HIV virus changed their structures very frequently, and the genetical difference and the mutations to change the genetical structure of the virus. So that is one of the most challenge. Second, our body not able to develop the natural immunity against the HIV virus. And the third, there is no particular animal models that can predict what will be replications in the human being. Okay. Our second question is, why would HIV drug resistance be issued in the era of ART scale-up? What could be the mechanism of the drug resistance? Whenever to suppress any virus or any bacteria, we need the minimum amount of the drug into the blood. That is known as a plasma level to suppress any bacteria or the virus. But many people who are not adherent, that means not disciplined in taking their medicines because of the one or another reasons, that in that times the blood in bloods there is a drugs, but that is not optimum levels. So whenever there is a suboptimal levels drugs present in the blood, the HIV virus can start to replicate, multiply, and the HIV virus can learn that how to resist or how to get the actions, how to immune from that or medicines. And during that process, and the HIV viruses multiply very fastly. In the AIDS patients that not are on the ART medicines, approximately 1 crore new HIV virus per day develop in that persons. So due to the suboptimal level of the medicine in our blood and the indisciplines, that is the main reasons for developing the HIV drug resistance. Why do HIV vaccine candidates develop so far failed to show a good protective efficacy during the preclinical and the clinical trials? Again, the same questions and same answers are there. The major 4 reasons are there. That means that we are failing to develop the HIV vaccines. First, there is a lack of natural immunity after the vaccinations also. Second, very highly multiplications of HIV virus within very short times. Till date, more than the 5 crores structurally different HIV virus exist in this earth. Third, there is no correlations between how HIV immunity will help for the futures by any models. And the fourth, there is no animal models that can predict our effectiveness of the vaccines in the human being.
Thank you so much, sir, for answering all the questions. That was the last question for today's session. And as of now, we haven't received any questions from the viewers yet. And at the outset, I would like to thank you, sir, for taking out your valuable time and for being here at our platform and sharing such wonderful inputs upon the topic on such an auspicious day that you have shared. So thank you so much for that. And I would also to thank all the viewers for being here with us and for connecting with us and for staying tuned with us throughout the session. So thank you to all of you. So with all your last comment, we can conclude the session here for today.
Thank you very much for watching these videos, and we will see with some other topics next time. Thank you.
Thank you so much, sir. Thank you. With this, we are concluding the session here for today. Until next time.
Okay. Thank you.